Anandamide is a lipid mediator that acts as an endogenous ligand for the cannabinoid receptor 1, CB1, in the brain, essential for signaling that regulates neurotransmitter release, particularly GABA, to manage neural excitability. CB1 receptors are primarily located in the central nervous system, brain and spinal cord, on presynaptic neurons, but are also found in peripheral tissues such as the liver, adipose tissue, and muscle. Anandamide takes its name from the Sanskrit word ananda, meaning joy, bliss, delight, plus amide. These receptors are also the primary molecular target responsible for the pharmacological effects of delta-9 tetrahydrocannabinol, the psychoactive ingredient in cannabis sativa. Several studies demonstrate that anandamide exerts a modulatory effect on the brain's reward circuitry. Several reports suggest its involvement in the addiction-producing actions of other abused drugs, and it can also act as a behavioral reinforcer in animal models of drug abuse. Importantly, all these effects of anandamide appear to be potentiated by pharmacological inhibition of its metabolic degradation. Enhanced brain levels of anandamide after treatment with inhibitors of fatty acid amide hydrolase, the main enzyme responsible for its degradation, seem to affect the rewarding and reinforcing actions of many drugs of abuse. Anandamide research shows it's a crucial endocannabinoid involved in mood, reward, pain, and appetite, acting on CB1 receptors to influence well-being, with low levels linked to anxiety, depression, and PTSD. At the same time, exercise and CBD may raise it, suggesting therapeutic potential for disorders like chronic pain and addiction, though their rewarding effects also hint at abuse liability. Studies explore boosting anandamide by inhibiting fatty acid amide hydrolase, FAAH, its breakdown enzyme for conditions from stress to appetite loss, but also reveal complex roles in reward, motivating drug-seeking in animals. The hormone oxytocin, which has been associated with interpersonal bonding, may enhance the pleasure of social interactions by stimulating the production of marijuana-like neurotransmitters in the brain. Scientists have provided the first link between oxytocin, dubbed the love hormone, and anandamide, which has been called the bliss molecule, for its role in activating cannabinoid receptors in brain cells, thereby heightening motivation and happiness. To investigate the role of anandamide in social contact, scientists measured levels of this marijuana-like neurotransmitter in mice that had been either isolated or allowed to interact. Anandamide is among a class of naturally occurring chemicals in the body known as endocannabinoids that attach to the same brain cell receptors as does marijuana's active ingredient, THC, with similar outcomes. The researchers discovered that social contact increased the production of anandamide in a brain structure called the nucleus accumbens, triggering cannabinoid receptors to reinforce the pleasure of socialization. When cannabinoid receptors were blocked, this reinforcement disappeared. Scientists looked for a possible connection between anandamide and oxytocin, which is well known for its role in promoting social contact. A small number of neurons in the brain make oxytocin and use it as a neurotransmitter. When the scientists stimulated those neurons, they saw an increase in anandamide creation in the nucleus accumbens. More importantly, they found that blocking anandamide's effects also blocked the pro-social effects of oxytocin, which implies that oxytocin reinforces social ties by inducing anandamide formation. It was also found that interrupting anandamide degradation enhanced the pleasure of social contact. Animals treated with a drug that stops anandamide degradation behaved as though they enjoyed spending time with their cage mates more than animals treated with a placebo, Piomelli noted. Oxytocin has also been called the hug hormone, cuddle chemical, and moral molecule due to its effects on behavior, including its role in love and female reproductive functions. Scientists revealed that oxytocin makes people feel more extroverted, and clinical researchers are investigating it as a possible treatment for the symptoms of autism. But it's very hard to deliver oxytocin, a small protein, to the human brain. Research findings on anandamide opened the exciting possibility that drugs that block the degradation of anandamide, which are currently being tested for various anxiety disorders, could give a boost to the brain's own oxytocin and help people with autism socialize more. Drugs that block the degradation of anandamide, an endocannabinoid, function primarily by inhibiting the enzyme fatty acid amide hydrolase, FAAH, which is responsible for breaking down anandamide in the brain and peripheral tissues. These agents elevate natural anandamide levels, leading to anti-anxiety, antidepressant, and analgesic effects without the typical side effects of direct cannabinoid substances.